Okay, so this is the second part of the basics video, and this is a diagram of protein synthesis. And right now I'm sure you're looking at it going, what is that? I have no clue what any of this is about. I promise you it's actually not that bad, and it will make sense to you. So the first thing I did was I looked at this diagram and thought, is there anything here that I know what it is? And immediately I looked at this thing here, and that is clearly a double helix. So I know for sure that that is DNA. Okay. The other thing I know is that DNA is only found in the nucleus. So I'm betting that this area here, this is the nuclear membrane that makes the DNA, makes the nucleus. So this, I'm going to go ahead and label this the nuclear membrane, okay, which is what is the kind of the border of the nucleus. So you can go ahead in parentheses and write nucleus, okay. Now, is there anything else I know here? I know I've got some DNA here, and I know it's in the nucleus. And I can also see, look, AAA on one side, TTT on the other side. So it's showing me my complementary base pairing. So the first step of protein synthesis, we know, is transcription. And transcription is when DNA is used to code for messenger RNA. So clearly what's happened here, along comes an enzyme, you can't see any hydrogen bonds there, it's broken them up, and what it's doing is it's using this strand here as a template to make a strand of messenger RNA, the single strand DNA. Now, complementary base pairing, we know A goes with T in DNA, but there's no T in messenger RNA, so along comes a U nucleotide, and it binds with that A, and so what we end up with, if you look here, a complementary strand of, and this is going to be messenger RNA. Okay, so AAA complementary base pairs, not with TTT, but with UUU. C, right? I'm trying to work this out. Oh, I'm reading it wrong side. AAA, and then the next one is G, C. Then I've got a C on the DNA. I've got a G on the messenger RNA. Then I've got a G on the DNA, and I got a C on the messenger RNA, and then I'm coming over to here, and I got an A. But because there's no T in messenger RNA, I have a U instead. Now, where I have a T on the DNA, I still have an A on the messenger RNA, because remember, the A is no different, right? It's like, you know, the A can complementary base pair with T if it's in DNA, but with U if it's in RNA, and then G. So this, if this side here of the DNA was the template strand, this is what we call a complementary strand of messenger RNA, and that just shows you transcription. We can see that the messenger RNA is leaving the nuclear membrane, so it's going outside of the nucleus, and it's going to this thing here, and this is called a ribosome. And ribosomes have all different sorts of drawings, right? But pretty much always they look like they've got, you know, it might be a big bit and a little bit, it may be like this, but it's always kind of that kind of shape, okay? And sometimes you just see it's like a shadowy blob. So here it is, UUU is over here, UUU. And then we've got CGC, CGC, and UAC, UAC. Okay, this was transcription. In the ribosome, we have translation. And translation is, okay, so 1, 2, 3 bases in DNA is called a gene. The complementary bases, so this would be here, this is a gene. The complementary bases over here in messenger RNA is called a codon. Okay? Now, codon sounds like a code, doesn't it? So UUU in this messenger RNA is going to code for something. What does it code for? I need to know this business here. Okay. This is called a codon chart. And the first letter is U. So I come over here. First letter is going to be in this box. Everything over here on this side, I'm going to get a bigger pen. Right? Everything in this whole section here starts with a U. Okay? Now, in the case of the one that we're talking about, the second one is a U. So I come up here, and these tell me all my second letters. So now, everything in this section here, right, is also a U. Okay? So it's kind of like that. Now, the third letter is also a U. So I come over here, I'll look where these intersect in this section here, and I say U is in this one here. So come along, and that top box there would be U, U. I know that sounded really confusing, and that's okay. We'll get a lot of practice at doing this, which tells me that UUU stands for this amino acid here called phenylalanine, or PHE. Okay? So this amino acid here would be PHE. Now, this thing here, this is a tRNA molecule. And here's what the job of a tRNA molecule. On one side of it, it's got not a codon, but what we call an anticodon. On the other side, it has a specific amino acid. So AAA on this side will always have PHE on that side. Right, this one here is CGC. Come back to my codon chart. First letter is C. So everything kind of in this section here, right, starts with a C. The next letter is a G. So I come up here, second letter's G. So everything in this box right here is CG, and then the third letter is a C. So come over here to CGC, and it's ARG, arginine. So that is the amino acid arginine. So this is going to bring phenylalanine, PHE, next to ARG. Okay? So then you come along with the next one. It's CAU. All right? So let's look. CAU. First letter C. Second letter A, which is there. So that's everything in this box. Third letter U is up there. Histidine. So this amino acid up here is histidine. So what's going to happen is histidine is going to come down, join there, and then it's going to join to this amino acid. And we'll end up with a chain of amino acid, joined to an amino acid, joined to an amino acid, which is our protein. Okay? So the sequence of genes controls the sequence of codons, and the sequence of codons controls the sequence of amino acids, and that is translation.